Welcome, science friends, to Scientific Method and Experimental Design. The overview of our 8 steps are observation, question, hypothesis, experiment, data collection, conclusion, retest, and communicate. Step 1, observation. Observation are based on senses or tools, our 5 senses: sight, smell, hearing, taste, and touch, and tools that we can use to measure different things. Perhaps a ruler measuring length, a scale measuring mass, or a thermometer measuring temperature. There are so many different tools that scientists use to make observations. Observations of natural events usually raise a question. So, looking at our example here of 2 plants, what's an observation you have about these plants A and B? I'll give you a moment. Some of the observations I made were that plant A is in the shade and is smaller than plant B, which is in the sun. Step 2, question. Your observations may lead to one or more questions. Some questions that I had are that, why is plant A smaller? Does the weather affect the plants? And is this due to predation? Is something eating our plants? Step 3, hypothesis. Based on your observation, a hypothesis is formed that tries to explain your observation or answer your question. A hypothesis is a prediction. We're trying to predict or determine the outcome of an experiment before we do the experiment. This is also known as a claim, if you are familiar with a CER, claim evidence reasoning. Predictions usually start with an if and have a then in them somewhere as well. So for example, if the plant on the right is in the sun, then the bigger plant is in the sun. A full hypothesis statement contains a because. So my example, here's my hypothesis. If plant B is in the sun, then the bigger plant is in the sun because the sun makes plants grow bigger. Step 4, experiment. Test our hypothesis. So our experiment will be testing our hypothesis. Was our prediction true or was it false? So an experiment is something scientists set up in order to test that hypothesis. Scientists tried to design experiments that clearly show whether a particular variable caused an observed outcome. So what is something that changed between our 2 things, or more than 2, that changed the outcome? Really important to note that if it cannot be observed, then it cannot be tested. So for example, can we test if a comet impact killed the dinosaurs? If so, why? If no, why not? I say no, we cannot test that because we can't observe it. We don't have a time machine. We can't go back to when dinosaurs were roaming the earth to see what killed them all. We can't test to see if it's a comet. We can't lasso a comet and pull it into the earth and cause this giant impact and kill things on earth. We can't do that. So that is not a good hypothesis because we can't test it. In our experiment, I would put plant A in the shade and plant B in the sun in order to observe any differences in growth. So is the sun going to have an effect on our plant? Step 5, data collection. Data is collected from experiments. We have to record those observations or measurements. There are 2 kinds of observations. We have qualitative observations, which are a description. We're using words to describe something that we're seeing, or hearing, or smelling, or whatever we're using our senses for. The other kind is a quantitative observation. This is some sort of number data. When we're using a tool to measure something, how big is it? How heavy is it? What temperature is it? How hot or cold? We use those tools, we use our senses to make those observations. When we collect our data, we need to remember record it so we can look back at it later to analyze it. We like to organize things in science in tables, charts, and graphs. That way we can look at it and see, okay, well, what's going on here? We can analyze that data. I have 2 examples for observations of our plants. We have qualitative and quantitative. A qualitative observation would be talking about their size. Plant A is smaller than plant B. A quantitative observation would be to take a ruler or a measuring tape to look and measure how big are these plants. So we're using numbers. Plant A is 10 centimeters tall and plant B is 20 centimeters tall. So we're looking at numbers. Step 6, conclusion. We collected our data, we analyzed our data, but what does it mean? Scientists decide whether those results of our experiment support the hypothesis. We can support our hypothesis, or we can refute or not support our hypothesis based on those results. It's okay if you predicted correctly. It's also okay if you did not predict correctly. There's no such thing as a failed experiment. All experiments have real observations of real things that are happening. If there are results, it's successful. So, a conclusion for ours would need to answer this question. Did the results confirm or refute the hypothesis? In my example, with my example hypothesis, I would have a confirmed hypothesis. Yes, we're seeing more plant growth in the sun. Step 7, retest. In order to verify our results, we need to do the experiment again, and preferably again and again and again and again and lots more times. This is to make sure that our results stay the same or constant. If we're getting the same results time after time after time, that's great news. That means we're setting up our experiment the right way, our changing factor, in this case the sun or the shade, we're really sure that that's the thing that's having an effect on our, in this case, plant growth. Step 8, communicate. Results can be communicated in the form of a written paper, an article, a journal, or sometimes even a presentation at maybe a science conference. Communication helps other scientists not only aware of the things you're studying and researching, but also if they're interested in running those experiments, how they can do that. If we get other scientists to do our experiment, that's awesome. That's so cool because they can run the experiment and we can compare our results. Are they seeing the same thing? Are they seeing different things? If it's the same thing, great news. If it's different things, what's different? How did we set up our experiments? Is it perhaps a location that has an effect on our differing results? It helps to see if it's repeatable, consistent. That's the scientific method in experimental design. I hope this helped.